Today, we're going on an incredible journey. But forget about sprawling cities or complex machines. We are shrinking down, way, way down, to explore a factory that is far more intricate and efficient. A microscopic metropolis humming with activity right inside every single one of your cells. We're about to tour the assembly lines that build the very molecules that make you, well, you.
Okay, let's dive right in. Inside each of your cells is this amazing, interconnected network of membranes and organelles that all work together. Think of it like a perfectly synchronized assembly line. This whole setup is called the endomembrane system, and it's the core production line we'll be exploring today.
So our first stop is to just get the lay of the land. Let's map out the different departments of this amazing cellular factory and see how they all work together. So on our factory floor plan, we've got the star of our show, the endoplasmic reticulum, or ER for short. That's the main manufacturing hub. Right next door is the Golgi complex, which is kind of like the post office of the cell. It sorts and ships all the products. Then you have specialized cleanup crews, like lysosomes and peroxisomes, that handle waste and hazardous materials. They all work together in this seamless, coordinated dance. Now, to give you a sense of scale here, check this out. The endoplasmic reticulum, our main character today, actually makes up more than half of the total membrane in most of your cells. It is a massive, sprawling network right at the heart of all cellular production. All right, so to really understand this factory, let's follow one product from start to finish. We're going to track the journey of a single, brand new protein as it's born on the factory's main assembly line, the rough endoplasmic reticulum, or RER. So why do we call it rough? Well, it literally looks rough under a microscope because its surface is covered with millions of these tiny dots. But they aren't imperfections. They're ribosomes. They're actual little machines that build proteins. And this is where our protein story officially begins. But wait a second. With all the chaos going on inside a cell, how does a new protein know it's supposed to head over to the ER for processing? Well, it's pretty cool. It has its destination encoded right into it, like a molecular zip code. This little address label is a short sequence of amino acids called a signal peptide. So as the very first part of the protein chain emerges from the ribosome, that signal peptide is immediately recognized. A kind of molecular courier, called a signal recognition particle, or SRP, latches right onto that zip code. And the moment it binds, it slams on the brakes, pausing all protein production. Everything just stops. Now, with production on hold, this SRP carrier escorts the entire complex. That's the ribosome, the messenger RNA, and the partially built protein, all the way over to the surface of the ER. It docks at a specific receiving station, the SRP receptor, and once contact is made, the SRP just lets go. Its job as a guide is done.
And here's the brilliant handoff. The ribosome is now locked onto the ER membrane, right over a special protein channel called a translocon, which is basically a gateway into the ER. The production brake is released, and as the protein chain gets longer, it's threaded directly through this gateway and into the interior of the ER, which we call the lumen. It's a remarkable bit of efficiency. The protein is being built and imported at the exact same time.
Okay, so our protein is inside the ER, but it's not done yet, not by a long shot. This raw protein chain is pretty useless as is. It now has to be folded, tagged, and pass a super rigorous inspection before it can be shipped out. The first step is getting the right shape. Specialized proteins inside the ER called molecular chaperones, like this one here, named BIPE, act like expert origami masters. They bind to that unfolded chain, guiding it, helping it twist and bend into its precise, functional, three-dimensional structure. Now, while it's being folded, the protein gets a really important modification. A complex sugar tag, an oligosaccharide, gets attached. This tag is actually pre-built on a special lipid carrier in the ER membrane called dolichol, and then it's transferred as a single unit onto the protein. You can think of it like a quality control sticker that's going to get checked later on. And let me tell you, this quality control is no joke. The ER is like a high-stakes inspector. Proteins that are folded correctly get their sugar tags slightly trimmed, which is the signal that they've passed inspection. But if a protein is misfolded, it gets retained, identified as faulty, and ultimately shipped out for destruction. And this isn't just about efficiency, it's critical for survival. When this system fails, the buildup of these junk proteins can lead to devastating diseases like cystic fibrosis. So we've been focused on the rough ER, the protein specialist, but it's physically connected to another region with a completely different set of skills, the smooth endoplasmic reticulum, or SER. The smooth ER, as its name suggests, doesn't have ribosomes, so it has a smooth appearance. Its whole world revolves around lipids. This is where the cell synthesizes the phospholipids it needs to build membranes, where it makes cholesterol, and where it creates the precursors for steroid hormones. And crucially, it's also the cell's main detoxification center. And this isn't just abstract biology. You see this in action all over your body. For instance, a liver cell, whose job is to detoxify your blood, is absolutely packed with smooth ER. The same thing is true for cells in the testes and ovaries, which need to produce huge quantities of steroid hormones. The amount of SER a cell has directly reflects its function. Okay, let's get back to our protein. It's been perfectly folded, it's been tagged, and it has passed that intense quality control inspection. It is now officially ready for shipping, so it's time to package it up and send it to the next station in the endomembrane system. You might be wondering, how do we even know this pathway exists? Well, for that, we can thank George Palliday and his Nobel Prize-winning experiment. It was a brilliant bit of scientific detective work. He fed cells a short pulse of radioactive amino acids, and then he could literally watch where newly made proteins went over time. The radioactivity first showed up in the rough ER, then it moved to the Golgi, and finally it was packaged up and shipped out of the cell. He mapped the entire journey for the first time. And this is exactly what that first step of the journey looks like. Our protein, along with some other cargo, gets packaged into a tiny bubble of membrane called a transport vesicle. This little vesicle then buds off from a specialized exit site of the ER and begins its trip along the cellular highway to its next stop, the Golgi apparatus, the cellular post office, where it'll be modified even further and sorted. You have now followed a protein's journey all the way from assembly to its very first shipping container. That was a ton of details, so let's see what's stuck with a few quick questions. First question. Right at the very beginning of our protein's journey, what was the specific job of that crucial courier, the signal recognition particle? And for our second question, let's shift gears away from proteins for a moment. Can you remember two of the specialized jobs handled by that other department, the smooth ER? And for a final thought to leave you with, this factory system is remarkably efficient, but it's not foolproof. The accumulation of misfolded proteins is a major source of cellular stress, and understanding how this intricate quality control system can fail is really one of the keys to understanding the origins of so many human diseases.